Hi, Dr. Crowder here. I'm making this video as a brief overview of bacterial genetics, specifically looking at bacterial conjugation, and then analyzing interrupted mating and recombination experiments to map genes in bacteria. And this video is particularly relevant to students enrolled in my BIS 101 course or other BIS 101 sections at UC Davis. So we're going to briefly go over conjugation, specifically focusing on conjugation between an HFR cell and an F- cell. And in this case, in the HFR cell, the fertility factor plasmid has been integrated into the bacterial chromosome, where the bacterial chromosome is indicated by teal and the fertility factor is the red region. And you can see that in the F- cell, the bacterial chromosome does not have the fertility factor integrated in it. More so, the F- cell does not have the fertility factor. And as we know, the fertility factor encodes genes that are necessary for building the pilus, which allows for conjugation. Now we want to focus in briefly on the black and the green region indicated in the bacterial chromosomes. For in the F- cell, it's the black region, and in the HFR cell, there's this green region. And these two regions indicate areas of homology, meaning that they're regions of the chromosome that encode for the same DNA sequence. So now let's examine what happens during conjugation. During conjugation, a pilus forms from the HFR cell that builds a physical contact with the F- cell. And then, starting at the fertility factor origin, the bacterial chromosome is replicated through a process called rolling circle replication, where replication begins at the origin, indicated by this arrow, and continues along the bacterial chromosome. And as replication is occurring, a single-stranded DNA copy is then transferred through the pilus to the F- recipient cell. Then, once in the F- recipient cell, that single-stranded DNA copy is replicated to form a double-stranded DNA molecule. And the result then is that part of the fertility factor, because the origin is in the fertility factor, and then the associated bacterial chromosome next to the origin is transferred into the F- cell. And if we remember then the regions of homology between the two bacterial chromosomes, one in the F- cell and one in the HFR cell, they share regions of homology, which promotes and allows for crossing over or exchange between the complete genome of the F- cell, called the endogenote, and the partial genome that was transferred over through conjugation, called the exogenote. And so at this current stage, the cell has two copies of one segment of the DNA. One copy is part of the endogenote and the other copy is part of the exogenote, resulting in what's called a partial diploid or meriploid. And if such a crossover event happened, where it's a double crossover or quadruple crossover, it results in exchange of genetic information between the two bacterial cells, where now the F- cell has integrated in the bacterial chromosome the DNA sequence from the HFR cell. And the remaining segments of chromosome will be lost in subsequent cell growth and division that were not integrated into the bacterial chromosome. So that is a very brief overview of bacterial conjugation, which is only one mechanism for how bacteria are able to exchange and take up genetic information. They can also exchange and take up DNA through transduction and through transformation, but those are not the focus of this video. However, we can use then this process of bacterial conjugation to map genes on the bacterial chromosome through interrupted mating experiments and recombination mapping experiments. Let's first walk through an example of an interrupted mating experiment. And in this experiment, we are mating an HFR cell that is prototrophic for Lu, Trp, and Arg and sensitive to the antibiotic kanamycin. And we're conjugating this with an F- cell that is oxytrophic for Lu, Trp, and Arg, but resistant to the antibiotic kanamycin. So this F- cell needs to have Lu, Trp, and Arg added to the media for it to be able to grow. So we take the HFR cells and the F- cells and we mix them together in the media. So now we have our tube that contains both the HFR and the F- cells in the same physical space, allowing for conjugation to happen. And then what we do at different time points, we're going to take samples from this conjugation mixture and plate it out onto different media conditions. And the first thing we want to do in our interrupted mating experiment when we plate these cells out is to select for F- cells, because these are the ones that are going to be receiving DNA through conjugation. And it is the time in which the DNA segments from the HFR enter the F- cells that informs how we are able to order the genes relative to the fertility factor origin. So in order to do that, when we take our time point samples from this conjugation mixture, we need to select for the F- cells only and therefore get rid of the HFR cells. And we do that by adding kanamycin to the minimal media. And if we remember, the HFR cells are sensitive to kanamycin. So adding kanamycin to the minimal media on the different plates that we plate these out on is going to specifically kill the HFR cells, allowing us to select for the F- cells after conjugation. Now, when we take our time samples and plate them out, we're going to plate them out on different media conditions. All of the conditions are going to be minimal media plus kanamycin. But then we're going to have different conditions where we are able to select for whether the F- cell has received the prototrophic marker from the HFR. So in the first plate, we are adding leucine and tryptophan to the media. So that means that the F- cells we are selecting for are the ones that have then received the Arg prototrophic marker, because the cells that grow in this condition are able to grow in the absence of Arg, which the F- cells alone cannot. So the only cells that are going to be able to grow in the absence of Arg are those cells that have received the prototrophic marker from the HFR chromosome that was copied and transferred over during conjugation. For the second condition, we are selecting for F- cells that have received the Trp prototrophic marker from the HFR, because we are not putting tryptophan in the media. So only the F- cells that have received that prototrophic marker through conjugation will be able to grow. And then in this third condition, we are selecting for F- cells that have received the prototrophic Lu marker from the HFR, because these cells would be able to grow in the absence of Lu. So again, we mix our cells and we allow them to undergo conjugation. And at different points after mixing the cells, we then take samples from this master mixture and plate it onto different media conditions. So if we do that, if at time 0 we take a sample from our master mixture and plate it out under these different conditions, we would expect no colonies to grow, because there hasn't been any time for conjugation or transfer of bacterial chromosome DNA from the HFR to the F-. Let's say we let some time go by and then take another sample. We take another sample and plate it out on these different conditions. So after 10 minutes, we now see that we have some colonies starting to grow in the absence of tryptophan, or Trp. And what this tells us then is that the prototrophic Trp gene was the first to transfer over during conjugation, meaning that it's the closest to the fertility factor origin on the HFR bacterial chromosome, so O for origin. So then if we wanted to map these genes relative to the fertility factor origin, we would say that TRP is the first one, or the closest. So now we're going to let some more time go by, 10 minutes later. So now after 20 minutes from the start of mixing these cells or allowing them to conjugate, we take another sample and plate it out under the different media conditions. And what we see now is that after 20 minutes, we see that cells or colonies are surviving in media that is lacking arginine. So what that tells us then is that the prototrophic arginine marker was the second marker to transfer over to conjugation. And so now we can order arginine as the second closest marker to the F factor origin. And now if we let a little bit more time go by, and 30 minutes after mixing these cells, we take another sample and plate it out under these different conditions, we now see that we have cells able to grow in the absence of leucine. That tells us then that of these three, leucine was the last prototrophic marker to transfer over during conjugation. So leucine is the furthest from the F factor origin. So by analyzing this interrupted mating data, we were able to deduce the order of these three markers in relation to the fertility factor origin in the bacterial chromosome of the HFR strain. So now that we've ordered our genes, where TRP is the closest to the origin and LU is the furthest from the origin, and I should write here that kanamycin, with respect to everything, is the furthest as well. We now want to map the genes relative to each other as far as the physical distances of the gene, or we want to create a more detailed map. So for example, if in our interrupted mating experiment, we wouldn't have been able to order the two of the markers because let's say the F- cell acquired the ability to grow in the absence of two markers at the same time point, we wouldn't be able to distinguish between the two. So recombination mapping allows us to fine-tune our map both with regards to order if the interrupted mating does not give us a clear order, but more so it allows us to map the physical distances between the genes. So here again, we are allowing our HFR and our F- cell to conjugate by mixing them together. And we're going to allow them to mix for an amount of time that allows for the region we are interested in on the HFR bacterial chromosome transferring over to the F- cell. Because what we want to ensure that the entire region we are interested in transfers over to allow us to be able to accurately analyze the recombination mapping data. So in order to do that, we need to plate our cells out on media that has kanamycin, again because we want to select for our F- cells only, so we're going to get rid of our HFR cells. And to select then for the F- cells that have received the entire region of interest, we are going to plate these out on media lacking leucine, so that only the F- cells that have received the entire area of interest that we are considering will be able to grow on media lacking leucine. So we're ensuring that this last prototrophic marker has transferred over by plating the cells out on media with kanamycin, therefore killing the HFR strain, and lacking leucine, selecting for only the F- cells where leucine has entered the recipient. After we do our first selection, selecting again for F- cells where the entire region has transferred over during conjugation, we then can replica-plate or replate out our cells onto different media conditions to then analyze the genotypes of the given X conjugates or F- cells after conjugation to determine the frequency of different genotypes that have resulted from crossing over. And to do this, we can replica plate where we have our master plate, which this plate would be the plate with can but lacking Lu, so our F- cells that have received the entire region we're interested in, we press that onto a sterile velvet that the bacterial cells will actually stick to, and then from that imprint of cells, we can then play on to different media conditions to then analyze the frequency of different genotypes in our population. So again, we play on media with kanamycin to specifically select for F- cells, and then we play on media lacking leucine to select for cells F- X conjugate cells that have received the entire region of interest, where we know that leucine is the last marker to be transferred over during conjugation. And then we replica plate on different conditions to analyze the different genotypes of those cells we've selected for that will give us information about the frequency of crossovers between the different markers. Where we know a crossover can occur between regions of homology on the endogenote and the exogenote. So we could have a crossover here, 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 and here to exchange genetic information. But remember that we can only have an even number of crossovers, or only those events where there is an even number of crossovers will be propagated. Because if we had an odd number of crossovers, so if there was just one crossover, if we track this crossover, so let's say starting here, over, and right, continuing this way, if we track this crossover and pull it out into a single line, you're going to have one linear piece of DNA. And in bacteria, DNA are not propagated during replication as linear DNA. They have to recircularize to be replicated and propagated over time. So therefore, we need an even number of crossovers to occur to maintain or recircularize the bacterial chromosome. So let's say we've done our recombination experiment. We've selected for the F- cells that have received the entire region of interest. We've replica plated, and then under the different conditions, we've counted the frequency of genotypes. And I'm giving you the frequency here. If we know the order of genes, which we established before as the trip is closest to the origin, and then we have ARG, and then we have LU. We can then take that information along with the genotype frequencies to map the genes by counting the frequency of crossover between the two intervals, or between genes. So if we look at this first genotype, where it's LU plus, ARG plus, TRP plus, since we already know the order of the genes, we can say that the only way F- recipient cells would have this genotype is if there is a crossover. We can number these positions, 3, 4. If there is a crossover at 1, so the bacterial chromosome exchanged LU plus, ARG plus, TRP plus, and then a crossover back again at 4. And right, the recipient event would be happening where on the exogenote we would have an exchange, so it would be transferring LU minus, ARG minus, TRP minus, and that linear piece of DNA would be lost. What we want to look for next in our recombination mapping data is the least frequent class. The least frequent class is the class where there was a crossover at every one of the intervals. So it was the 4 crossover event. And we could imagine that having 4 crossovers is going to be a lot less frequent, or it's going to happen a lot less than having 2 crossovers. So in the least frequent class, there's a crossover at 1, so you get LU plus, 2, ARG minus, 3, TRP plus, 4. So we have 4 crossovers, resulting in this genotype LU plus, ARG minus, TRP plus. And if we didn't know the exact order of all of the genes, we could look at the least frequent class that has 4 crossovers and determine the gene order, where the Rh minus, or the one that's in the middle, is going to be the one that is different than the other two. So it's the auxotrophic marker that's different from the other two. And that one, for the least frequent class, has to be the one in the middle because that's the only way 4 crossovers would result in that genotype. And so now we want to take our data from a recombination experiment to map the distances between the genes. And so we first want to look at the distance between Lu and Rh, and then the distance between Rh and Trip. To map the distance between Lu and Rh, we want to look at the frequency of crossover between those markers. And so that would be this class and this class. So if we look in this first class, it's Lu plus Rh minus. So to result in that, we would have to have a crossover here, Lu plus Rh minus. So crossover at 1 and 2. That's the only way we would have the phenotype Lu plus Rh minus Trip minus. And we already established that the Lu plus Rh minus Trip plus is the result of the 4 crossovers. So there was a crossover at every interval, including between Lu and Rh. So then to map the distance between Lu and Rh, we would add up the frequency of the crossovers between those two. So 14 plus 1 divided by the total number. So it would be 0.15 or 15 map units. Now, if we wanted to map the distance between Rh and Trip, we would look at the classes where there was a crossover between those two, which again would be the 4 crossover class and this class. Where this class, the only way you could result in that genotype would be a crossover at 1, Lu plus, Rh plus, and then a crossover at 3. A crossover between the Rh and the Trip minus. Okay? So then we would add those up, 26 plus 1. This 1 again is coming from the 4 crossover, where we know there had to be a crossover between Rh and Trip, divided by the total, so 27 map units. So now, by a combination of the interrupted mating and recombination experiment, we have our order, where if this is the fertility factor origin, we have Trip and Rh, and we know that they are 27 map units apart. And then Lu is the furthest marker from the origin, and the distance between Lu and Rh is 15 map units. So hopefully this gives you a further understanding of how we can take advantage of the process of conjugation to do interrupted mating and recombination experiments that allow us to both to determine the order of genes on bacterial chromosome as well as the map distances between those genes.